decay milk. Microorganisms that can decay fossil, no,
That cause
decay,
decay.
or that cause decay,
Yeah.
bacteria,
Yes, bacteria and
bacteria, and
fungi.
yeah, fungus.
Hmm.
Name the product that causes the pH to decrease. One of the products are the breakdown of lipids, the amino acids. As
Uh,
milk decay, lipids in the milk are broken down. One of the products of the breakdown of lipids causes the pH of milk to decrease. Name the product that causes the pH to decrease.
yeah, it is fatty acids. Okay. Fatty acid cause it to.
Give one way the pH can be measured. pH probe.
Yeah.
I give two control variables a student should have used in the investigation. So, look, test tubes. To each test tube to enter the same cow's milk.
Yes.
Use the same cow's milk and same goat's milk. Record the number of days they can
Volume of milk should
control.
also be same, right?
Yeah.
Hmm.
Type of milk stays fresh the longest at 10 degrees. So at 10 degrees, the almond milk, no goat's milk.
Goat's milk pH is
No, no,
what?
no. What's the? Is it cow's milk?
Let me see. Cows is this line. Then goat. Almond milk, right? Because almond milk is this. The time taken to reach pH 5 is
Oh,
pH 5 is here. This is pH 5.
describe the effective temperature on time taken for goat's milk to reach pH 5. Um, goat's milk is that one. So until 15 degrees, no, from 30 degrees to
Hmm.
what?
So as the temperature increases up to 15, as the temperature increases up to
As temp
15 degrees,
increases to 15 degrees,
the time taken,
the time taken
hmm,
decreases
and as the temperature
until
increase above 15, time taken to reach pH 5 stays the same, right? You can see this constant graph.
so after 15 degrees where it stays constant
Yes.
at 2 time taken for cow's milk to reach pH 5 at 10 degrees is less than time taken for cow's milk to reach pH 5 at 5 degrees. Uh, because when a higher temperature um,
Because enzyme
temperature
activity is faster at
means enzymes are faster
as at 10 degree as compared to
at
5
10 degrees
because there
compared
is more kinetic energy.
time. So there's two reasons why the different types of milk take different lengths of time to reach pH 5. They all They have different starting pHs.
Okay, yeah, yeah, they have different, and different milk have different static pH. So, and
Yep.
then different milk could have different concentration of fat and lipids in it, right?
Has different concentration of fat.
Yes, or lipid.
Since as the temperature milk is stored at affects how likely the milk is to cause food poisoning. How can the investigation be developed to find out if the student's correct? The temperature of milk stored at use more than three different types of milk. Record pH. Determine types of bacteria present in the milk. Determine types of bacteria present in the milk.
Yes, yes, that's correct.
Calculate how many times greater the human population was in the year 2000 compared with the year 1900. So in 2000, the population, well, one sec. So 2000 population is
Hmm.
5.9.
No, it's 6
So
billion.
5
In 2000, you
6
can see it's
billion.
crossing this line, so it's 6 billions.
6 billion. And then in 1900, it is one sec, 1,
Yeah,
1.2, 1.4,
this
1.6, 1.8.
1.6, right? If you see carefully, 1.8 is this point, but this is 1.6,
Is
isn't
it
it?
not? Wait, 1.2, 1.4. Okay, 1.6.
Yeah. So you will now 6 billion and 1.6 billion. What you will do now? Divide it, right? 6 divided by 1.6.
So 6 divided by 1.6 equals 3.75 billion.
Yes.
Well, it's 3.75 times more,
Y-
I think.
Uh, yes, 3.7. Yeah, it is times. So 3.75 times more.
In 1950, human population was 2.5 billion. Calculate the mean annual increase in the human population between 1900 and 1950. So from 1900, it was 1.6 billion,
1900, yeah,
billion,
1.6 is given. Okay.
and then it
Nine.
went to 2.5 billion.
Yes.
Then 2.5 minus 1.6
Yes. Subtract
equals
them.
0.9.
Okay.
This this increase 0.9. So then mean annual increase.
So now you will divide it by 50 years. The difference between 1900 and 1950 is 50. So divide that difference of 2.5 and 1.6 by 50.
So 0.9 divided by 50.
Yes.
0.9 divided by 50 equals 0.018.
Yes, that's your answer. Very good.
Predict the human population in 2050 if the current rate of population increases continuously. You should draw an exponential line figure to predict human population 2050 if the current rate of population increases. So
Now you will
pick a
do,
random.
just extend this graph, okay? Just take it like this. Let me do it here. Because as you Prediction is that it will be going up because as you can see, as it is growing exponentially,
Do I just use the ruler?
like this. This is the graph, okay? Draw it like this, okay?
Do I use a ruler or freehand?
No, no, freehand. Using ruler, how would you do it? It's exponential, right? Curved. Just use your hand. And if you have to do 2050, right? So
Just in the middle.
yeah,
So
this.
2050, I got 10 billion.
Yeah, okay, write it down. 10 billion.
Predicted investment 10 billion. The increasing human population has caused a decline in fish stocks. Describe how fish quotas can help fixing fishing quotas can help to return fish stocks to a sustainable level. Increasing the fishing quotas
Yeah,
means more
because yeah,
fish
remaining fish
is
can
caught.
reproduce.
What?
Catching fewer fish, that means remaining fish can reproduce, right?
So reducing quo- quotas means less fish caught, which reproduce more.
Yeah,
Farming techniques have changed in recent years. Describe why more land is being used as farming, how increased farming has decreased biodiversity, and increase more land is being used for farming is being
yeah,
used for farming as demand has increased.
yeah, as in,
Demand for goods has increased
yeah,
population increases. Population increases.
the
So
demand for food also increase.
more farmland required to grow crops. Uh, this means less biodiversity
Yes, because crops are consist
as
of
lot
plant that are of same type. So same type plants means little, it means very little biodiversity.
uh, land full of biodiversity removed in order to create space for crops. This means
Deforestation.
less
Hmm.
habitats and deforestation,
Which reduce biodiversity.
extinction, which reduces biodiversity in the area. Genetic modification of crop plants can help meet the demands of increasing the human population. Golden rice is genetically modified. What is the advantage of golden rice compared to non-GM rice? Golden rice contains protein-rich maya. Golden rice improved nutrition. Golden rice is genetically modified crop. Is it is improved nutritional?
Yeah, yeah, that's correct. This one.
Improved nutritional.
Yes, it improved nutritional value.
So there's one reason why some people are concerned about the use of golden rice. It is not natural,
Yeah,
so is
also
unethical to
it may enter
unethical
in
to
other species. It may enter in other species, or it reduce traditional wild rice, or it, okay, it also reduce biodiversity. Okay, you can write any one reason.
this this question about plant hormones. Farmers can spray seeds or give gibberellins to start germination. What are the two uses of gibberellins? To increase fruit size
Two
and
boxes. Okay.
to promote flower production.
Yeah, to increase fruit size. Yes, that's these two are correct. Okay, great.
Describe two improvements a student should make to their investigation. Student investigate how the effect of light intensity on the height of seedlings.
Hmm.
There are two improvements. Um,
They should keep temperature same for all of the seedlings, right? And use
the
equal number of
temps
seedlings.
for seedlings
Use
use
equal number of
same
seedlings in each dish.
flower seedlings.
Yeah, this will create a fair investigation. Okay.
Suggest how students measured the length of the curved seedling using a ruler.
Using a ruler, length of curved seedlings. Um,
They can just use a ruler.
yes, I think. E- or it should be, you should write flexible ruler, okay? With a flexible ruler so that we can bend it. Or or we can write tape measurement. Tape, there is a tape which we can which is flexible, right? Because it is curved, it is not straight. If we have a straight ruler which is not flexible, we cannot use it. So write flexible ruler.
Explain what happened to the growth of seedlings on side Q compared with the growth on side P. Oh, the auxin in the shoot of the seedling moved to side P
Hmm.
because the because it was attracted by the light
Yes, exactly. Light.
for growth. So side Q doesn't grow because
The auxin
it
causes.
has no light
Hmm.
on it. So side P curves towards light source.
Yeah.
Bananas are often stored separately from other fruits because bananas release a plant hormone. Why does storing bananas with other fruits cause the other fruits to ripen faster? Bananas contain um,
Hmm. Because bananas release ethene. Okay.
bananas release ethane.
Yeah.
Why is DNA described as a polymer?
Polymer means
Uh,
something which is made up of many monomers. Monomers means single units. Poly means many. So DNA is
polymer
made up of many monomers, and these monomers are
chain
called nucleotides.
monomers which in DNA are called nucleotides.
Yes.
Describe the structure of a nucleotide.
Yeah. So
I know nothing about nucleotides.
so let me write. Nucleotide is made up of a phosphate group and a sugar. Let me show you here.
Nucleotide means phosphate group
Phosphate group is attached to a sugar
sugar attached
and sugar is attached to base.
and sugar attached to base
Base will be one of four possible bases A, G, C, and T.
of four bases A, C, G, T.
And C, yeah. Okay. So these three things combine phosphates, sugar, and base, and they make nucleotide.
Okay. The length of a DNA double helix increases by 0.3 nanometers for every pair of nucleotides. The total number of nucleotides in the human body is 1.2 times 10 to the power of 10. Calculate total length of the human body cell using information from figure 5.
Yeah.
So, length of double helix increases.
So,
So, I'm assuming, so is the
hmm,
double helix in the human body cell is 0.34.
nanometer.
That's the starting.
Yeah.
What's the whole of the double helix though?
0. Let me show you here. So as you can see, it is given 0.34 nanometer. This is the width.
Oh, that's how it increases though.
Yeah.
That's
So
for every pair of nucleotides.
yeah, for every. This is fixed. So total number of nucleotide is given, which is this. And for now, let me
So
see.
that 0.34 nanometers to meters.
Yeah, we'll convert it into meters.
Is that 10 to the power of 8? Or
9. It will be 9. Let me just open the door. There is someone at the door. One second. Assalamu alaikum. So, yeah, I'm here. Let me go back to the question. So
do
it
I
is,
divide by 10 to the power of 9?
yes, you have to divide by 10 to the power
So
9.
it's 0.34 divided by 10 to the power of 9 equals 3.4 times 10 to the power of minus 10
Okay, 3 point,
meters.
3.4.
4 times 10 to the power of minus 10.
Okay, yeah.
So then you do 3.4 times 10 to the minus 10 times 1.2.
No, because 1.2 is total number of nucleotides in a human cell. We have to calculate total length of double helix in a human body. So this is what we need to do. This is a one
Divide.
yeah, we have to, this is for we have to 1.2, if we divide it by 2. Okay, we have to divide it by 2 because we have to find the total length of double helix, and this is given for cell, for the total number of nucleotides in a human body cell. Okay, so if we divide 1.2 by 2, what we will get? 6, right? Point
One
like,
point. So why is it divided by two?
because this is given total number of nucleotide, but we are finding total length of double helix. So if we double it, like this is, suppose this is a single strand, if we double it, this becomes half, right? This length, right? That's why we are dividing it by 2. Okay,
So the number of nucleotides is two times what the double helix is.
yeah, we yes, we have
Um,
to divide this by 2 because this is for 1, and if we divide, the length will become half.
helix length. So do I do 3.4 times 10 to the minus 10 divided by 0.6 times 10 to the minus 10 to 10?
No, you are not supposed to 0.6 times 10 to the power 10. This is what you have, right? Times
Yeah, do I
it.
divide
Now you are
uh,
supposed to times it with uh
0.6 times 10 to the power 10 times 3.4
yes. I think
times
that
10
was
to the
negative 10, right? 10 to the power negative 10.
0.6 times 10 to the power 10 times by 3.4 times 10 to the negative 10 equals 2.04 meters.
Yes, very good. That's correct answer.
Some parts of DNA do not code for proteins. Describe how non-coding parts of DNA can affect the expression of genes. Non-coding parts of DNA can affect the expression of genes.
Hmm.
I have no clue what that means.
Okay.
Some parts of DNA do not code for
Only
protein.
some parts code that we can see in expression which express their effect, but non-coding parts of DNA, they can switch genes, and that's how they can affect the expression of gene if they switch genes.
So what's the answer?
Non-coding parts of DNA can switch genes or
Non-coding parts of DNA can switch
on or off.
genes on or off.
Yeah, this is their function. They can make genes on and off.
Okay. Uh, there are two types of cell division: mitosis, meiosis. Meiosis has four daughter cells,
Yeah, meiosis produce four daughter cells, produce,
whereas four daughter cells,
and mitosis produce two, right?
mitosis only produces two.
Hmm.
Meiosis daughter cells have 23 chromosomes each.
Yeah, or you can write set the daughter cell in mitosis are diploid. Diploid means two set of chromosomes.
So
Mitosis.
daughter cells in meiosis are diploid,
Mitosis,
but
okay?
in mitosis diploid.
Yes. And meiosis is haploid.
So haploid, diploid, and then
Mitosis produce identical cells
mitosis produces identical cells.
and meiosis produce different cells.
The meiosis produces different cells. Describe one similarity between the process of mitosis and meiosis. They both have daughter cells.
In both of them, yeah, DNA replication take place for copying, and uh, they copy by DNA replication, right?
I just said they both produce daughter cells.
Yeah.
Give the genotype of person one. Dupuytren. Is it
Let,
Dupuytren? So is it D, capital D?
okay, let me explain this. I think
I don't
person
get it.
one. Okay, it is affected, and yeah, capital D. It can be capital D, or capital D, small d, right? In both case. So, but now let's understand through the offspring if which one is this. So let's see this one. You can see if I take it as capital D and small d, and the other one is d small d, because it is unaffected, right? Capital D means affected
Yeah,
and small d means unaffected. So
yeah,
now these two are also unaffected, right? And if we cross
yeah,
these two are affected, right? Because see, one, if you cross it with this and if you cross it with this, so it is going to be capital D and small d, okay?
yeah.
Now reason you will write, um, it has a big D because they have um, uh, dipterines, it but must have small d to, because like to, so that their offspring.
Wait, don't. What? Why is it have dominant allele?
Let me write answer here. So the answer is this has a big D because they have two twins but must have small d because they have offspring that are D and D, right? Because these two are affected. To make them affected, this should be the genotype of one, right? That are.
Person 7 and person 8 in figure 6, expecting a fourth child. What is the probability of the child having Dupuytren? So person 8 is without, so he's DD.
Okay, let me go here. Person 8
So
is small d, and person 7 is capital
is
D and small d. Now we cross it here, okay? This is
D.
person 7 and this is person 8. Capital D, small d. Here again you will write small d and small d because person 8 is unaffected. Now cross them. capital D, small
D.
d. This is DD.
So capital D, small d, D, D,
These two.
and then D, D,
So
D,
these two, they are
thing.
affected.
Identify the probability is 50%.
Yes, probability is 50%. You can see 2 out of 4. 2 fourth, which is 50%. Okay.
Explain how figure 6 shows how the allele for deuteriums is not on the Y chromosome.
Yeah, because if you notice here, female 3 and um, in female, where is this?
Uh,
Hmm. So write down female 3, 11 and 12. 3, 11 and 12. I'm talking about these two. They have, they are affected,
are affected by
and females do not have Y chromosome, right? Y
but
chromosome.
females don't have Y chromosome.
Yeah, that is the reason.
Okay.
Let's look at sixth.
Figure 7 shows the brain. Which part of the brain becomes more active if a person balances on one leg? Um, C is pituitary gland, I think. So not that. Is it A or
Yes,
B?
it's A. This is cerebellum. Okay, B is medulla oblongata, C is pituitary gland, and D is cerebrum. So the answer is
Okay,
A.
so name the part of the brain that's responsible for making the decision. D.
Making a decision. Yes, it is D. Cerebrum. Okay, so
Wait, so the answer to 6.1 was A.
yes, it is A. 6.1
In
is A.
most MRIs, in most MRI scanners, the person being scanned needs to stay completely still. A functional MRI scanner allows a person to move whilst the scanner makes images of the person's brain activity. Suggest how the functional MRI scanner would help to find out more about the brain damage a person has. It
Yeah,
can tell. It
so you can, yeah.
can it show discrepancies from how a normal brain would look
You
when
can,
moving.
yeah, you can ask person to do a specific task to see which part of the brain is active or inactive. You can
The ask
compare
person
this.
to do task to see what part of brain specifically is inactive
Or active, yeah, you can compare this to a person without brain damage to be able to
and
identify where the damage is.
to person without to identify. Describe how the brain receives
Okay, I
information about
think
light and things.
we can continue in next class. Okay.